Let's start with a biology lesson about what RNA is and what types of processes it's involved in. So you may remember from your high school biology course that RNA is an essential molecule for all of life, and it participates in numerous biological processes. You probably learned about transcription and translation as part of the central dogma of biology. In transcription, DNA, which stores the information for your cells, is turned into a messenger RNA, so a form of a copy of the DNA. You need the DNA, of course, to do that. You need RNA building blocks called nucleotides, and you need an enzyme that creates that RNA copy of the DNA. That's transcription. That transcribed messenger RNA leaves the nucleus and then eventually winds up in the ribosome. And in the ribosome, its job is to translate that messenger RNA to produce a protein. And proteins are the doers of the cell. So the information from the DNA makes its way out and is turned into the doers of the cell, the protein, via this messenger RNA vehicle. And that is actually how these COVID vaccines that people are so familiar with nowadays, they're built upon that idea, basically. The message for the spike protein that is present on the SARS-CoV-2 virus is encoded in a messenger RNA. A patient receives a dose, and the patient's ribosomes translate that messenger RNA into the spike protein. And that is what allows the patient's body to be trained against the COVID virus should one come into contact with it. Your immune system picks up on that spike protein and learns to not like it. So you probably learned about transcription and translation in high school biology, but one process you may not have learned about is RNA interference. So in that process, double-stranded RNA, so a different type of RNA now, can be chopped up into smaller RNAs, and these small interfering RNAs can bind to messenger RNA as a way of targeting it for degradation. So what that means is, let's say you had a protein and you didn't want to make that protein anymore with that message, you can send little RNAs to bind to that message so that that message gets chopped up into bits before it can actually make the protein. An example of a solution that we've been working on that leverages RNA interference is for the Colorado potato beetle, a pest for potato plants. So this long double-stranded RNA that we make is present in liquid form, and it's applied by the farmer to the leaves of the potato plant. So it's a spray. It's been designed in a way such that it fits with the farmer's regular regimen of applications, so he doesn't have to do anything special or pick up any special applicators. It was designed to work with things that he's already used to doing. He sprays it on his potato plants. Along comes the beetle. The beetle's eating the leaves as part of its natural life cycle, and it ingests this RNA. And the RNA gets in there, and like we talked about a minute ago for RNA interference, it targets an essential message for that beetle that's necessary for making a critical protein for the beetle, and it knocks out that message. So if the beetle needs the protein, it no longer can make it. And that is how we make the beetles sick. They're not hungry anymore, and they eventually die. What's also great about RNA, though, is it's sequence specific. So it's been designed, and it will work against the beetle, but it will generally leave things like birds, bees, butterflies alone because the sequence was not designed to target them and only the beetle. The other great thing about RNA in terms of this application is it is a natural molecule, and microbes in the environment will naturally degrade it. So it is biodegradable due to the nature of the molecule, so it does not have long environmental persistence. And all of these things are what make double-stranded RNA particularly interesting for use in the farmer's toolbox. This chart shows a timeline for how we have developed that first solution for Colorado potato beetle. Starting over on the right, we have filed with EPA for approval of that product already, and we think that it will be registered in 2022. But if you rewind the clock back into late 2016, we had no infrastructure, no experts in field trials, no experts in beetles, no experts in how to make it. What this is really meaning to illustrate is that in just 6 short years, we've gone from a concept all the way through to filing this first molecule and anticipating regulatory approval in 2022. In 2017, we filed some IP, which was ultimately awarded. In 2020, we have a US patent that protects our ideas for making these long double-stranded RNAs. During the course of this timeline, we also developed analytical methods, a formulation. We chose the target sequence that we needed that does the doing in the beetle. We put together a field trial operation. So we did 29 US field trials during 2019. We expanded that to go international, including the EU and Canada in 2020, and we did over 60 field trials across those geographies. And we did an additional 40 last year between the US and Europe. So we put together a very large field trial program to get all the data we needed to show that this product was efficacious and safe. We also designed, built, and are now operating a pilot facility in Rochester, New York, where today we're capable of producing up to 0.5 metric ton of this material as we prepare to ultimately get the solution registered and we start producing that molecule. One other point to make on this slide is that we didn't want to be a one-trick pony. We acquired some IP from Bayer in late 2020, and we intend to file that product with EPA also in 2022, so in a very short period of time from when we picked up that IP. Beyond that, we didn't even want to have one or two products. We knew that we needed to build an engine that would set us up to produce numerous other products. So at the same time, we were developing the solutions for Colorado potato beetle and acquiring the one for the bee health product. We also knew that we wanted to bring other solutions in other areas, fungal, insect, plant health, et cetera. And we knew that we needed to build an engine to drive all of that product development. What we set up essentially was a classic design, build, test, learn loop where we design the sequences using bioinformatics, algorithms that we have in the computers. We put together infrastructure that allows us to produce hundreds of sequences per week, and then we can test those hundreds of sequences to see which ones are performing the best in terms of being an efficacious solution and a safe solution. And naturally, through the course of testing those in our bioassays, we arrive at sequences that are more interesting. And those ones we can further iterate on. We can go back to them and make slight tweaks and improvements to them so that we can improve them even more before we gate them for late stage development. Assuming we get candidates that meet requirements for commercialization, we then will take them out of that research and discovery phase and we'll put them into full-blown development and scale-up. And as I mentioned, we have this facility in Rochester, New York that today is even capable of producing up to 500 kilograms a year using our platform process. And that is what we can use to satisfy global field trial programs across all the product development cycles. So imagine you're a scientist and it's 2016 and the CEO comes across and knocks on your door. He says, hey, what do you think about double-stranded RNA as a potential use of our technology? He knew we had been looking at things like nucleotides and complex biocatalysis and energy regeneration and all of these things. And he thought, it fits the mission. It fits, where we are with the technology. What do you think about it? And so you say, okay, interesting. Tell me more. Can you tell me how long would these RNAs have to be? And he says, pretty long, hundreds of base pairs. So not real short. And why that matters is short RNAs you can make with synthetic chemistry, but longer ones you can't because the longer the RNA gets, the more error-prone the synthetic approach goes. So right away you can kind of scratch the synthetic chemistry approach off of your list for how one would do this. And so then you say, well, okay, we're not going to do it with chemistry, so we've got to do it with biology, enzymes. How much do we need to make, and how inexpensive does it have to be? And he says, well, for these agricultural products, we're going to need metric tons to cover the acreage. And by the way, the active ingredients got to be cheaper than, say, $1 per gram. And so you take a big gulp and you think, well, okay, interesting. Let me think about this. So that sort of rules out the way that the messenger RNA companies were thinking of approaching things. They use a technology called in vitro transcription, and it's infeasible for hitting that type of scale or that type of cost. For a reference point, think about the COVID vaccines today. Those processes are on the order of kilograms, not metric tons. And the cost for a 50 microgram dose, let's say, is probably on the order of $1 to $10 per dose. And here we're talking about making 1 million times as much and putting it in a jug for the same cost. So it causes you to, again, gulp and think a little bit differently about how one might do that. So what you're left with essentially is a process called fermentation, right? You ask the cells to make the RNA for you. The catch with that is microbial cells that you'd use in fermentation, well, they don't like to make a lot of RNA, and they're going to fight you. And you can say, well, we'll reprogram and we'll make them better at it. Sure. The issue then that you have is that they do not want to accumulate a lot of RNA. And so they've got mechanisms in place to tear down that RNA. And what that means for you practically, if you're trying to develop a commercially viable process, is that your yields are low and the quality is going to suffer. So you say, well, all right, fermentation also has some issues. So let's think if there's a better way. And so the way that we devised to do this was thinking outside the box a little bit. So we knew we needed inexpensive raw materials for making our RNA in order to hit this metric tons and low cost per gram. How can I get my hands on some readily available, inexpensive RNA building blocks, nucleotides? And thought, well, where is all the RNA in nature? It's mostly present inside of cells, and specifically in the ribosomes inside the cells. So where can we get some readily available, inexpensive ribosomes that we can use as our raw material? And, we were aware, obviously, that in the food industry, the brewing industry, there are a lot of yeast-based processes in the world. And so these yeast cells, after they're done doing their job, they've got RNA left in them. we can get the RNA from the yeast cells and use a special enzyme called a nuclease to degrade that RNA back into individual RNA building blocks. Voila, we have our RNA building blocks for making our RNAs for our plant health solutions. The issue with that is that the building blocks themselves need to be in a certain energy state. So the building blocks that you get after you digest the yeast RNA are called nucleoside monophosphates, 1-phosphate. They need to actually be triphosphates, three phosphates, higher energy state, in order to be polymerized by the enzyme that turns them into RNA. And so we also needed a cost-effective way to do energizing of these nucleotide building blocks. Good news for us, we had been thinking about that for several years, and we were aware of an enzyme, an inexpensive source of essentially high-energy phosphate called polyphosphate. That's nothing more than old-school water softener. So our building blocks for making RNA are really nothing more than yeast and then old-school water softener to serve as the energy source. So you don't just need that, of course. If you remember back to transcription in biology, you need the DNA template. You also need some enzymes to read those DNA instructions and convert those building blocks into the RNA. So we had to figure out how to make those DNA instructions and make those enzymes as well, in addition to making the nucleotides. We spent some time and we developed processes for those. The one interesting thing that we did along the way there was we knew, based on how difficult it was to make RNA in fermentation, that there were going to be enzymes present as impurities in some of these materials that would degrade our RNA, that would degrade the DNA that we need to make the RNA, that would degrade the enzymes we need to make the RNA, that would degrade the nucleotides that we need or modify the nucleotides that we need to make the RNA. All of that is bad news for us if we want to make a lot of RNA on the cheap. And so the solution we came up with for mitigating all those undesirable activities that you don't want in your enzyme or template preparations is heat. So we sourced enzymes from thermophilic organisms. All that means is these organisms grow at really high heat. Those enzymes, those proteins, they can survive high temperatures. So we knew if we produced enzymes and used them in our process that could survive at high temperatures, that we could do those in microbes that grew at normal temperatures. And when we were ready to prepare them for use in our process, we would just turn up the heat and essentially cook away all of those unwanted activities that we didn't like, the ones that degrade the RNA, the DNA, the enzymes, the nucleotides. We leveraged thermostable enzymes in our process. That was critical for us as well. Of course, we have to recover the RNA as shown on this graphic, and ultimately, depending upon the solution that you're going after, whether it's in a beetle or another insect or in a fungi, your formulation is going to vary from product to product, but making the actual double-stranded RNA active ingredient for each solution more or less follows this process. So we had developed a process for making double-stranded RNA for plant health solutions. And naturally, we had cobbled together a tremendous amount of know-how, a lot of technical expertise in different areas in order to bring that to fruition. We had to figure out how to make and manage the nucleotides, the DNA, the enzymes that you use to make the RNA from the nucleotides and the DNA, how to recover that RNA, how to analyze all of those things with analytical methods. So all of that tremendous amount of know-how and expertise, a logical place to apply it next was to making messenger RNAs for human health type solutions. And the catch there is that messenger RNAs are a little bit different than these long double-stranded RNAs. Messenger RNAs are generally longer. They also require modifications at each end of them. At one end, they require what's called a cap, and at the other end, they require what's called a tail. So you had to think about adapting the double-stranded RNA process for making this much longer single-stranded molecule where the ends are modified in certain ways. Not to mention the fact that these messenger RNAs need a delivery vehicle in order to get into the cells so they can do their doing. The technology that one uses to do that, at least today, is called a lipid nanoparticle. So the process had to get familiar with making these mRNA lipid nanoparticles for use in our solutions. And finally, this material is going to be administered to patients. And so demonstrating control of purity and GMP compliance is of the utmost importance. So the analytical methods, the data systems that we had, the quality systems that we had in place, we had to step them up even to another level. And we also had to be aware of some of these messenger RNA-related impurities that weren't present in our double-stranded RNA process and how we would manage and control those as part of the product.